Hello students, so today our topic of discussion is cell determination. Now before we start with cell determination, let us discuss how a multicellular organism develops. We know that a multicellular organism, it develops from zygote after repeated mitotic divisions. When I say mitotic divisions, it means that all the cells, they are genetically similar. Genetically similar means they have all the cells have the same amount of genetic material in them. Now, if all the cells in our body, they have the same genetic material, then how some cells become liver cells, some become blood cells, some neurons and some muscle fibers? Why only the liver cells, they secrete the bile juice, and why hemoglobin, it is produced only in the red blood cells? So maybe some genes, they are expressed in certain cells but are turned off in other cells. So let's study the cell determination and find out whether it is true or not. So what is cell determination? Cell determination, it is the process by which the nuclei of the cells of an embryo at some stage of the development, they lose the potential for full genetic expression and their fate becomes fixed. It means that as the development proceeds, जैसे जैसे development stages आगे बढ़ती जाती हैं, वैसे वैसे उनका जो full genetic expression का जो potential है वो कम हो जाता है और उनकी जो fate है that becomes fixed. A German embryologist, Hans Drisch, he worked on sea urchin and he observed that the blastomeres of sea urchin up to the 4-cell stage, that is after the second cleavage, till first 2 cleavages, they are totipotent. Totipotent means that they have the full potential for development into a complete embryo, right? So we can say that these blastomeres are not determined yet. Still their fate is not fixed. They have the potential to develop into a complete embryo. Such cells are called totipotent cells, right? Similarly, another scientist, that is Hans Spemann, he worked on frog and he observed that the blastomeres at the 2-cell stage, that is till the first cleavage, after the first cleavage, they are totipotent. Again, totipotent means they have the full expression, they have the full potential to develop into a complete embryo. So as the further cleavages take place, their fate becomes determined or fixed. After 2-cell stage in frog, the fate of the blastomeres, it becomes fixed. And in case of humans, these cells, they become determined after 3rd cleavages, right? After 3rd cleavages means by 8-cell stage, till the 8-cell stage, the cells are, blastomeres are the totipotent, right? So if you separate the blastomeres after the first cleavage, then you will get the twins. And if after 2nd cleavage you separate the blastomeres, then it will lead to multiple births. Now why it happens? What is the limiting factor? So here the limiting factor is not the genetic potential of the nucleus but the cytoplasmic constituents. क्योंकि जैसे जैसे zygote डिवाइड होता है, ब्लास्टोमियर्स बनते हैं, तो वी नो कि न्यूक्लियाई तो डिवाइड हो जाता है, विद ईच ब्लास्टोमियर अ न्यू न्यूक्लियाई इज प्रोड्यूस्ड. न्यूक्लियस तो डिवाइड हो रहा है बट साइटोप्लाज्म जो है उसका अमाउंट इंक्रीज नहीं हो रहा. द सेम अमाउंट ऑफ साइटोप्लाज्म जो कि zygote में था, नाउ इट इज़ बीइंग डिस्ट्रीब्यूटेड इनटू डिफरेंट ब्लास्टोमियर्स, राइट? So the limiting factor is not the genetic potential of the nucleus but the cytoplasmic constituents. And as the cleavage takes place, the number of nuclei increase but the volume of cytoplasm, it becomes progressively reduced in each blastomere, right? So the function of the nucleus, it means it is dependent upon the cytoplasm, right? So now we will discuss the control of the cell determination. So cell determination, it is controlled by two factors, that is intrinsic factors and extrinsic factors, right? Intrinsic factors कौन से होंगे? जो cytoplasmic constituents हैं. तो cytoplasmic constituents, they are also known as ooplasmic determinants, right? Ooplasmic determinants which are distributed unequally between different blastomeres during cleavage. And what are extrinsic factors? Extrinsic factors means which act from outside, जैसे कि signals हैं between different blastomeres, आपस में signals pass on करते हैं, या due to position of the blastomeres उनको अलग अलग instructions मिलती हैं, and this also leads to cell determination, right? So now we will discuss the regulation of cell determination by ooplasmic determinants. So ooplasmic determinants basically क्या होते हैं? Ooplasmic determinants, they may be the proteins which regulate transcription, or they are the localized mRNA molecules. mRNA molecules, which mRNA, you know mRNA का क्या function है? It helps in protein synthesis. So localized mRNA molecules which are translated during embryogenesis. During embryogenesis means कि before fertilization, these mRNA molecules which are present in the cytoplasm of the egg, they are masked. Masked means they are inactive. So after fertilization, these mRNAs, they become active and they undergo protein synthesis, right? So after translation, they form some gene regulatory proteins. तो ये जो gene regulatory proteins हैं, these are called ooplasmic determinants, और इनका function क्या है? They regulate the differential gene activity. Differential gene activity means some genes they are turned on and some genes are turned off. So due to differential gene activity, they produce functional differences in the regions of the egg cytoplasm. तो egg cytoplasm के जो different regions हैं, उनमें different proteins distributed होते हैं, so they behave differently, right? So now there are two types of the regulation of ooplasmic determinants. एक तो germ cell determination होती है, एक somatic cell determination. So today first we will discuss the germ cell determination, that how the ooplasmic determinants, they determine the formation of germ cells कैसे बनते हैं. We know the germ cells, they are involved in reproduction, right? तो germ cells कैसे बनते हैं? सबसे पहले क्या होते हैं? Germ cell precursors, right? So these germ cell precursors, or we can say the raw material required for the formation of germ cells, they are known as germ cell precursors. तो ये precursors क्या बनाते हैं? They form the primordial germ cells. These primordial germ cells then migrate to the gonads and they multiply to form the germ cells, right? Let's see the next slide. So in this, first we will take the example of the germ plasm of the nematode eggs. So nematodes, for example, Ascaris. So the existence of the germ plasm, it was first demonstrated by Boveri in Ascaris eggs, right? So Boveri in 1887, they worked on Ascaris eggs and they found that there are two types of cells, that is somatic cells and germ cells. Now he observed that the cells which undergo chromatin diminution क्या होता है कि they lose some specific parts of the chromosomes. Some specific parts of the chromosomes, they are lost, and these कौन से parts lose करते हैं? These are actually the repeated DNA sequences, right? So only the structural genes are retained. So in Ascaris, the cells which undergo chromatin diminution during early cleavages, they form the somatic cells. The chromatin diminution में क्या हुआ? Some specific parts of the chromosome are lost and only the structural genes are retained and they behave like somatic cells. On the other hand, the cells which retain the full set of chromosomes, right? So here, just a minute. So the cells which retain the full set of chromosomes, they formed the germ cells. So two types of cells, germ cells and somatic cells, right? तो germ cells, why do they retain the full set of chromosome? अब सिर्फ इन्हीं cells में ही क्यों full set of chromosomes आया और somatic cells में chromatin diminution क्यों हुआ? Because ये जो germ cells हैं कहाँ से बनते हैं? germ plasm से, और ये germ plasm कहाँ पे present है? In nematode eggs ये germ plasm present है in the vegetal half of the zygote, right? तो vegetal half में cleavages take place in such a manner that only the vegetal cell they contain the complete set of chromosomes, so that a complete set of chromosome may be passed to the next generation. क्योंकि ये जो germ plasm है हम कर रहे हैं कि ओप्लाज्मिक डिटर्मिनेंट्स कैसे डिटर्मिन करते हैं, है ना? तो हम डिस्कस कर रहे हैं इसमें germ plasm. तो हाउ डू सम सेल्स दे गेट द कंप्लीट सेट ऑफ क्रोमोसोम्स व्हाइल द अदर्स, उनमें क्या हो रहा है? सम पार्ट ऑफ द क्रोमोसोम्स इज बीइंग लॉस्ट, क्योंकि द प्लाज्म और द germ plasm विच इज़ प्रेजेंट इन द वेजिटल हाफ ऑफ द zygote, तो उस germ plasm से जो germ cells बनेंगे ओनली दे कंटेन द फुल सेट ऑफ क्रोमोसोम, राइट? देन कम्स द another example is of insect eggs. So insect eggs may be, there is a pole plasm, right? So what is a pole plasm? It is a specialized cytoplasm which is present in the posterior pole of the insect eggs, and this posterior pole पे जो cytoplasm है it contains the germ cell determinants, right? So this pole plasm, since it contains the germ cell determinant, so it will give rise to the pole cells, and these pole cells then they give rise to the germ cells. So this pole plasm it undergoes cleavage and it produces the pole cells. तो pole cells क्या है? Here you can see in this diagram, this is the diagram of an insect blastula, and you can see on the vegetal pole there are pole cells, right? So what are these pole cells? These are actually the precursors of the primordial germ cells, and they will ultimately form the germ cells in the gonads. So now let's see in the next slide. So that was germ cell determination, and now we are going to discuss about the somatic cell determination, right? So in this we are going to take the example of tunicates. So you have already studied in second year what are tunicates. These are the ascidians, these are the urochordates. They have a tunicin, tunicin की एक इनके ऊपर एक लेयर होती है जिसको बोलते हैं हम tunic बोलते हैं, right? So that is why they are known as tunicates. These are ascidians. So in early tunicate cells, the fate of the cell, that whether this cell it will become a germ cell or somatic cell, or whether it will form which type of structure in the body, so that is determined by a specific type of cytoplasm which is acquired during cleavage, right? तो हम tunicates में example ले रहे हैं Styla partita की. Now in this case the different regions of the cytoplasm they have different colors. You can see in the diagram also there are different colors. So these regions which have different colors they become localized or they become separate like this. इस तरह का एक arrangement इनमें कब आता है? After fertilization. In the unfertilized egg of Styla, the yellow cortical cytoplasm it surrounds the gray yolky inner cytoplasm. So here in this first diagram you can see that the yellow colored जो part है, this is the this will form the future myoplasm. It surrounds the inner gray yolky inner cytoplasm. Inner cytoplasm it will ultimately form the endoplasm, right? So now during meiosis what happens? This is the nucleus. The nucleus of the fertilized egg. जब ये थोड़ा सा enlarge हो जाता है तो इसको हम बोलते हैं germinal vesicle. You can see here I have labeled it as germinal vesicle. Now during meiosis what will happen? This germinal vesicle it undergoes the breakdown, right? So the breakdown of nucleus it releases a clear substance that accumulates in the animal hemisphere of the egg. This is the animal pole of the egg, this is the vegetal pole of the egg. So when this germinal vesicle or the nucleus it breaks down, so it produces a clear substance which get accumulated in this animal hemisphere. This is the area, you can see this is the clear area. So it contains the clear cytoplasm here, right? Now in the next diagram, you can see that a male pronucleus is present. Male pronucleus kahan se aaya? By the entry of the sperm. So after sperm entry in the vegetal hemisphere of the oocyte, so this is the vegetal side. So from here the sperm enters. When it enters in the vegetal hemisphere of the oocyte, then the yellow cortical cytoplasm and the clear cytoplasm, it moves vegetally towards the sperm nucleus. So here in this diagram you can see that this yellow myoplasm, it has come here in the vegetal pole. Plus this clear cytoplasm, it also यहाँ से from here it starts migrating towards the vegetal pole, right? So after the sperm entry into the vegetal hemisphere, now the myoplasm, which is yellow in color, and the clear cytoplasm, they start moving towards the vegetal hemisphere or the vegetal pole of the egg, right? So as the now what happens in the next diagram, here you can see now the male pronucleus, it migrates from the vegetal pole to the equator of the cell, right? This male pronucleus, here you can see that it is migrating towards the equator of the cell. So as it moves from the vegetal pole to the equator, the yellow cytoplasm, that is myoplasm, and the clear cytoplasm, they move with it, right? So first, now they will move towards the equator, right? So the yellow cytoplasm, it moves into the subequatorial region, right? ये कहाँ पे move करता है? Subequatorial region में move करता है, and it forms a crescent in the posterior region of the egg. Crescent means, let's see in the next slide, the diagram, it will be clear to you in the next slide. Yes, so here you can see that the yellow cytoplasm, it has come here, right? So this yellow cytoplasm is here, and so it comes to lie in the subequatorial region, right? And the clear cytoplasm, that is ectoplasm, now it has shifted towards the animal pole, and a new cytoplasmic region, the chordoplasm, appears in the interior vegetal region. So this is the chordoplasm. एक नया cytoplasmic region develop हो जाता है in the interior vegetal region, which is known as chordoplasm, right? So these movements of the cytoplasm, they are called the ooplasmic segregations. So you are seeing what we are doing till now. You can see that if I show you the previous diagrams, just a minute. If I show you the previous diagrams, so you can see that in this diagram, so here you see initially the myoplasm, it was present on the periphery or in the cortical region, and then it shifted towards the vegetal hemisphere. Both ectoplasm and myoplasm, they moved in the vegetal hemisphere. So in this diagram you can see, right? And in the next diagram what happened, that they came to lie in the equatorial region, right? So in this region and in this region. So now we have the ectoplasm on the animal hemisphere, endoplasm on the vegetal hemisphere, chordoplasm in the interior vegetal region, and myoplasm in the posterior vegetal region. So these cytoplasmic movements, or they are also called ooplasmic segregation. So they are occurring simultaneously with the cleavage. Simultaneously the cells are dividing also, and ooplasmic segregation is also taking place. And this ooplasmic segregation, or the movement of the cytoplasm, it establishes the bilateral symmetry in the egg, right? So cleavage is also taking place. So this cleavage, it then distributes the color cytoplasmic regions into different blastomeres. So here you can see in this diagram that in this diagram you see this is the way, right. So here you can see, so this is the dorsal side, I have written wrong, and this is the actually the ventral side, right. So I will shift it like this. So this is the dorsal side and this is the ventral side, this is the posterior side and this is the interior side. So on the dorsal side, the cells containing the ectoplasm are present and they will form the epidermis, and the cells containing the endoplasm, they will form the gut, and the cells having chordoplasm, they will produce notochord, and the cells containing myoplasm, they will form the tail muscle. So here you can see in this diagram tail muscles, notochord, epidermis, endoderm. So this is how so this is how the ooplasmic determinants, they determine the different, how the different parts of the body are developed from different blastomeres. So let's see. Now the next topic is regulation of the cell determination by extrinsic factors. Till now we have discussed about the intrinsic factors. उसमें हमने discuss किया how germ cell determination takes place, how somatic cell determination take place, and now we are discussing the regulation of cell determination by the extrinsic factors. So extrinsic factors अब कौन कौन से हो सकते हैं? It may be either the position of blastomeres in the embryo or cell to cell interaction. So for example, if we take the example of mammals, then in mammals the 8-celled embryo, अगर हम stage study करें, so you can see there are two types of blastomeres, upper blastomeres and lower blastomeres. So here the position of the blastomeres, it will determine the fate of the cells, right? Because we are discussing the regulation of cell determination by extrinsic factors. तो यहाँ पे हमने mammals की example ली है जिसमें कि the fate of the blastomeres is determined by their position, that whether they are present on the outer side or on the inner side. For example, in the 8-celled embryo, which consists of 4 outer and 4 inner blastomeres, right? So the outer blastomeres, these are the outer blastomeres, so they give rise to trophoectoderm. And the inner blastomeres, they give rise to the inner cell mass. So we can see from this diagram, so after 8-cell stage, then comes the Morula stage. In Morula stage, you can see this is the subzonal layer. Subzonal layer, it will ultimately form the trophoectoderm. And this is the inner cell mass, which will give rise to the embryo proper, right? And this is zona radiata. So you can see in this side, in this diagram, that this is the inner cell mass and outer layer trophoblast, and then there is zona radiata, and then there is albumin. So the fate of the blastomere, it depends upon their location inside or outside of the embryo, and this hypothesis is known as inside outside hypothesis, right? So if we separate the blastomeres at 2-cell stage or 4-cell stage or 8-cell stage, so if these blastomeres, when separated from the other blastomeres at 2-cell, 4-cell or 8-cell stage, and if they are placed on outside of the mass of the blastomeres from some another embryo, हमने एक और embryo ली, उसमें से हमने जो inner mass of the blastomeres हमने लिए और उसके ऊपर हमने क्या रख दिए, blastomeres derived from the 4-cell stage, 2-cell stage और 8-cell stage. तो जब हमने उनको inner mass of blastomeres के outer side में रखा, then it gave rise to trophoectoderm, right? So trophoectoderm, so the cells which lie on the outer side, they give rise to trophoectoderm, right? So let's see. So it means that the cells which lie on the outer side of the embryo, they form the trophoectoderm in mammalian embryo. It also shows that the blastomeres at the 2-cell stage, so it also shows that the blastomeres at 2-cell stage, 4-cell or 8-cell stage, they are totipotent. Totipotent means they have the full potential to develop into any tissue or complete embryo, right? So that's all about the cell determination, and if you have any doubt you can ask me by writing in the chat box. So don't forget to like, subscribe and share it. Thank you so much.